because we are now on our final crash course for your biochemistry class. This topic is also very important because it appears in your board exam. So for crash course 10, we will going to highlight your different techniques in the laboratory, in particular for biochem experiments. So let's see. For this crash course, expect that I will discuss different enzymatic digestion techniques, your classical analysis for protein and nucleic acid separation and purification, and then thirdly, ayan, more advanced biochem techniques, your electrophoresis, PCR, and blotting techniques. So diba, strata tayo dyan class during our undergrad biochem. And fourthly, we have your brief discussion ng ELISA or your enzyme-linked immunosorbent assay. And finally, class, of course, don't forget your recombinant DNA technology. Class, let's first discuss our laboratory techniques overview. Alright, so class, again, for this discussion, here's a brief overview of your laboratory techniques. So class, in general, even in your other laboratories, Gen Chem, or Chem Lab, Phychem Lab, of course, we have similar techniques for isolation, identification, and characterization of your biomolecules. So, diba, for your isolation, we also call it your separatory technique or your purification strategies, okay? And of course, these techniques are integrated in order to fully elucidate the structure and determine the functions and unique properties of our target molecules. Alright, so class, I will flash some relevant equipment that you typically see in a biochem lab, okay? So I hope some of you might be familiar or sana familiar kayo, diba, with this equipment. So of course, here, class, can you guess this equipment? So actually, this is an ELISA workstation. So of course, ELISA, that is your enzyme-linked immunosorbent assay. So class, majority of your assay tests are actually done inside your biosafety cabinet, diba? So if I'm not mistaken, we have your BSc level 1 for, I think, non-pathogenic organisms, like your E. coli, diba? For our experimentation. And of course, until BSc level 4, for your life-threatening pathogens niya. So of course, those are in your advanced biochem lab, or mostly nga nasa clinical lab na siya for clinical testing. Okay, so class, second one, sir, nakalagay na yung name eh. Yes, so of course, very straightforward. This is your PCR. Class, here, very small ang PCR tube. And later, we will discuss the components of a certain PCR tube upon amplification in your PCR equipment. Alright, so for our third equipment, what is this kaya class? Of course, this is used for your electrophoresis, diba? So this is our source of voltage, and later we will discuss the specific mechanism and your theory that is very important for the electrophoretic experiment. And lastly, ayan. So of course, this is very familiar for all of us. This is your typical liquid chrom, or for our biochem, of course, we have your fast protein liquid chromatography. Class, again, this is just a teaser of what we are going to discuss in this crash course already na. So of course, we will start with the basics first, your classical or your wet lab biochem techniques. So let's start with your enzymatic digestion. So your class, clearly, we are just going to use your enzymes in order to degrade your large polymers, your biomolecules, diba? Carbohydrates, lipids, your proteins, and nucleic acids. Again, this is a classical characterization technique, and here we are just going to use your enzymes because they are specific to the biomolecule that they degrade or cleave. Let's check. Starting with your carbs. Sir, familiar yung iba? Yes, because we already discussed this from our discussion on biomolecules. Let's check. First, amylase again is to degrade your starch into its maltose component. Class, again, this is an initial degradation step. So of course, your maltose kasi is a disaccharide, right? So class, for your lactase naman, it will degrade your lactose into its monomeric units, glucose and galactose. Alright, for your sucrase and invertase, ayan, class, recall, what is your sucrose, diba? Reducing or non-reducing? So we know that this is a non-reducing sugar. See, its monomeric units are your glucose and fructose, and in your sucrose, they are joined together in their respective anomeric carbons. So class, naaalala pa ba? I hope wala kayong amnesia ha. So lastly, we have your cellulase. This is to degrade your cellulose. Remember, this is only present in your ruminants or your ruminant organisms. Of course, the monomeric unit of your cellulose is glucose. So diba? Very straightforward. Class, you need to remember this specific enzymes. Alright, so for our lipids, yan. Very familiar na rin because we discussed this in your lipid metabolism, particularly si pancreatic lipase, right? So here, we have your dietary tags, for instance. And of course, our product upon hydrolysis via your pancreatic lipase activity, of course, glycerol plus, diba, remember, two free fatty acids pa nga lang eh, and then we are also going to use your monoacyl glycerol lipase. Alright, so class, remember the specific monomeric units of your triglycerides. Next, for phospholipase, this is responsible in cleaving or in degrading the ester bonds in your phospholipids. Sir, parang nasa did yan? That's right, diba? Your snake venom are actually rich in phospholipase. Okay, and then lastly, si cholesterol esterase, specific siya in degrading your cholesterol esters to liberate our source of energy, diba? Your fatty acids plus cholesterol. Clear? Let's proceed with our last two biomolecules. Ayan, so class, very important. Ayan, I would like to emphasize that you are required to memorize the reaction mechanisms of these three enzymes under our protein. Again, sa trypsin, as we have discussed from our enzyme discussion, ayan, it specifically cleaves at the C-terminal of your basic amino acid, in particular, lysine and arginine. Alright, for chymotrypsin class, again, aromatic residues, your phe, trip, and tyre. And lastly, si pepsin, it cleaves particularly the hydrophobic residues in low pH. Clear? Again, these are your three main enzymes that can degrade or metabolize initially our dietary proteins kasi class. Okay, and then lastly, we have your nucleic acid. Check. So DNA is 1. This is to initially degrade your DNA into smaller fragments. Class, remember, our DNA structure is actually a complex one. That's why the degradation of our DNA double helical structure is a step-by-step, diba? Step-by-step process. So here, again, DNA is 1 will initially degrade your DNA into smaller fragments for access of your exonuclease and endonuclease. Second, RNase A. Sir, pang RNA yan, yes. From the name itself, it will relieve your single-stranded RNA, particularly at your pyrimidines residue or pyrimidine residues, your cytosine and uracil. And then lastly, of course, we have your exo and endonuclease. Class. Remember here, when we say exo, that's the terminal, okay, or the ends of your DNA and RNA, all right? So, of course, it cannot live within the DNA structure or your smaller fragments of DNA because it is sterically hindered. So, of course, that's why we have your endonuclease. Ayan, that is very specific to your endonuclease. Again, it will recognize your specific sequences and lives within your DNA strands. Clear? Ayan. So, class, very straightforward ang enzymatic digestion. And again, class, please remember the reaction mechanism of these three enzymes, trypsin, chymotrypsin, and pepsin, and of course, the difference between exonuclease and endonuclease. Okay, so class, let's proceed now with our another type ng classical analysis. O check your manual. There in your manual, we actually allotted a specific or a special topic for the protein and nucleic acid classical analysis because in the laboratory, well studied ang proteins and their activity, particularly their catalytic activity through molecular docking. And of course, it is in tandem with our instrumental analysis, diba? Your electrophoretic technique and as well as your classical analysis for, ayan, purification strategy class. Always pong classical ang ating separation or isolation techniques here. Or even man gumagamit tayo ng equipment or your glasswares, of course, it's still technically a wet lab technique. Okay, so let's check first with our protein classical analysis. So mainly, we use here your chemical digestion. Class, this is not limited lang to your enzymes. Again, we will use now wide range of chemicals in order to break your proteins into its corresponding peptide chain for sequence analysis. Class, here is the summary. First one, we have your Edman reagent. Class, this is a board exam relevant topic. Here, class, you need to remember the specific compound that we use in our Edman degradation, and that is your phenylisothiocyanate. But we will not discuss further the mechanism of binding of your PITC to your N-terminal amino acid. So actually, that's just a condensation process kasi. Okay? So again, to summarize, for Edman degradation, our reagent is your phenylisothiocyanate to sequentially remove your N-terminal amino acids. Okay? So meaning, we will sequentially remove each amino acid every turn. So actually, class, this is a tedious type of lab technique. So putin agad yung mata mo, hindi ka patapos, di ba? Lalo na kung 100 to 200 amino acid residues yung peptide mo. Okay? For our next batch of reagents, this tree labels your N-terminal amino acid. So class, remember, we have three chemicals here. Sildensyl chloride, C2,4-dinitrofluorobenzene, or it is also abbreviated as DNFB. And finally, we have your 1,fluoro-2,4-dinitrobenzene, or also known as your FDNB. Class, they are not specific. They can label all N-terminal amino acid. Clear? Again, remember, both your PITC and these three compounds or your chemicals can label N-terminal amino acids. Any amino acid, basta 20 standard amino acids. And lastly, for our cyanogen bromide, your CMBR, of course, will cleave specifically your C-terminus at your methionine residue. So class, if you have a copy of 2019 boards, it is actually a 2019 board exam question. But there, it is actually your analysis type of question, not definition type. That's why you need to know the mechanism or the function of your cyanogen bromide. Class, again, specific siya sa C-terminal ng methionine. Okay? So meaning that's on the right side of our polypeptide chain. All right. So class, in addition, we also have your different strategies in order to isolate, purify, or target our proteins. So of course, this is still a classical type of analysis. Let's see the first one. We have here your ammonium sulfate precipitation. Class, here, our basis of separation is through fractionation and purification. And here, we will use mainly your salt, ammonium sulfate, in order to alter the solubility of your biomolecule, particularly your proteins, to selectively precipitate them. Okay? So class, very easy in theory, see ammonium sulfate precipitation. But in the laboratory, it's actually tedious. Lalo na yung waiting time, di ba? Mabobore kayo. All right. Second, class, don't worry, we will discuss this in detail on our succeeding slides. But for now, let's have an overview of your differential centrifugation. Again, for our basis of separation rather, it's by size and density. Okay? So here again, sequential lang po ang centrifugation. So I expect that majority naman nakahawak na ng centrifuge, right? So here again, for differential centrifugation, in order to isolate our specific target biomolecules, we are going to sequentially expose them in different speeds. All right? So lastly, I think this is second to the last pala. Ayan, your IEF. Diba? Your isoelectric point is a characteristic pH of your proteins or amino acids in particular where they do not have a charge, okay? Or they do not have that net electric charge, which is equal to 0 lang siya. That's why here we have a specific strategy with its basis of separation by isoelectric point. So class, when we say focus, we immobilize your protein at their isoelectric point. So of course later we will discuss the theory in your IEF. And lastly, we have here your SEC or your size exclusion chromatography. All right, class, our basis of separation here is merely on their molecular size or weight, okay? So our column here is solid through your porous beads, and of course our concept here or the important concept that you need to know is that larger proteins will elute first because they will not interact that greatly with our porous beads, but the smaller ones will enter this pores and will have a brief interaction or weak interaction, and hence they will elute at a later time. Again, the first to elute, large proteins or large biomolecules, the last to elute because of their great interaction with the porous beads. Of course, that's your smaller proteins. Okay, so class, let's now discuss in detail each of this protein classical analysis. Starting with your ammonium sulfate precipitation. Class, very cheap ang purification strategy na to because we are only going to use your ammonium sulfate. Again, our basis of separation is solubility, and of course, here are the steps. Ayan. First one, of course, here is your test tube containing our dissolved target solute. So it can be a protein or any biomolecule. Alright? So class, mainly proteins ang ginagamitan ng ammonium sulfate precipitation. Second, ayan, we will now proceed with our salting out method. So class, here, we will just add your salt incrementally because of course, you can expect that there will be nucleation upon addition of enough salt. And of course, we don't want to over add your salt here because of course, it will affect the solubility of your other biomolecules present in the sample. Okay? So our third step, ayan, incubate muna natin. And then there will be a nucleation that will occur due to supersaturation upon addition of your ammonium sulfate. Class, here in your incubation step, this actually encompasses the nucleation and your particle cluster aggregation until we see your precipitate or your solids in our sample. And of course, afterwards, ayan, we will now centrifuge this test tube in order to concentrate our precipitate. And lastly, we are now going to collect your precipitate and your supernatant will be removed. Again, class, our key principle here is that your salt reduces your protein solubility, hence a selective precipitation process occurs. Alright, so let's proceed with our next step or technique. So class, I hope that you can follow, ha. So for those that did not experience this in your laboratory or undergrad biochem lab. So it's okay, class. So I assure you that I think 5 items maximum will appear in the board exam about this. So 5 or less, because again, majority of your items will be coming from your cellular respiration, biomolecules, and your other pathways. Okay? So don't overthink that much. Again, class, I'm going to emphasize naman the topics that are very sure na ako na nag-appear siya sa board exam. Alright, for our next discussion again, this is your differential centrifugation. And basis of separation again is your size and density. Okay, again, for our steps, I will flash our visualization here. First one, homogenization. So this is for your cell lysis. Again, we will first break down the structural property or integrity of our cells. Diba? Here, your cell homogenate will be suspended in a fractionation buffer. And this will induce broken cells or of course degradation ng cells. And here, we will liberate your subcellular components. Diba? So of course, after your cell homogenization, we will now we will now put it in your centrifuge at low speed first. Okay? So class, here, we are going first with your low speed to your higher speeds on our later centrifugation step. Check. Class, upon centrifugation at low speed, let's check what precipitated out. That's your whole cells. Of course, the first one to precipitate even at low speed are those large molecular weight biomolecules. Okay, so of course, upon or after your centrifugation, we will remove your precipitate in order to repeat your centrifugation step at higher speeds. And of course, during our last centrifugation, of course, it is expected that the smallest biomolecule will be the one or will be the last one to precipitate. Okay, so again, class, the important trend lang here is that in your differential centrifugation, the first one to precipitate out of your solution is those large or high molecular weight biomolecules. And the last one, of course, those are your smallest or your small molecules which has low molecular weight. Okay, so class, again, our key principle here is that the sequential spin sediment particles by mass. Okay, so I hope that's clear. Again, the heaviest and largest components will precipitate first, and typically this means your nucleus forms the first pellet after low speed centrifugation. Clear? Okay, so class, with our third protein classical analysis, this is your isoelectric focusing. Okay, so let's see. Again, our basis of separation is your PI or your IPH or your isoelectric point. And of course, here we need to have an input of electric field in our pH gradient gel in order to induce the mobilization of our amino acids in our gel. Okay, so after applying your electric field, your amino acids or in general the proteins will be focused at their PI via migration. And remember, diba, our amino acids have distinct PI values. See, here when we apply your electric field, they will migrate. So for low pH, the protein that is positively charged will migrate to your low areas or your low pH concentration, and the reverse or the opposite is true for your high pH. Okay, so class, afterwards, ayan, if we again apply a stronger electric field, your protein will start to migrate toward its isoelectric point. And upon reaching their isoelectric point, they will stop. Okay, they will stop migrating at your isoelectric point. That's why we call it your protein focusing. We are immobilizing your proteins at their PI. And then lastly, ayan, ready na for visualization ang bands natin. So of course, class, similar with your age, if you have conducted gel electrophoretic experiments, the very crucial step here is your sample pipetting. Class, this figure is just another visualization of your IEF. Check here. First thing is that an ampholyte solution is incorporated first into your gel, like this one. Of course, when we say ampholyte, those are molecules that have acidic and basic properties, like your amino acid. So here, a stable pH gradient is established in the gel when we apply your electric field. And of course, here, class, your protein solution will now be added and your electric field, will be reapplied. See? So of course, the first one is to standardize our pH gradient. Diba? From your anachem, always your standardization comes first. And lastly, ayan, after staining, you can now see the distribution of your proteins along our pH gradient with their PI values. Actually, class, IEF is in tandem with your gel electrophoresis, and we also treat this as your 2D gel electrophoresis because after these bonds have been formed, they will now be subjected to your gel electrophoresis for further separation. Okay, so I hope that's clear. Again, for isoelectric focusing, this is a lab method that separates your proteins based on their isoelectric point or the pH where your protein has no charge or no net charge. Okay, so class, let's proceed with our last protein classical analysis. Again, our main basis of separation here is your molecular size. Let's check. So parang yung typical column lang siya, class. Okay, typical chromatography setup. First, here is your injection ng sample on your column. So usually for our biochem, this is your FPLC. So of course, ayan, column injection. And then here we have your column. So we have your porous beads inside, and your proteins will be diluted and separated by their molecular weight. Okay, so sometimes our detector here is our spectrophotometer, and here we are detecting its absorbance at 280 nanometers. Okay, so of course, this will be plotted in your chromatogram. And lastly, second to the last, of course, we are now going to collect our fractions of interest. So actually, class, we also integrate this through our centrifugation in order to get our purified protein. And of course, ayan, if we are done with the purification, we will store it before we analyze it with using our different strategies. Diba? Very straightforward on size exclusion. Again, all you need to know here is that the basis of separation is its molecular size. And large molecules elute first, your small molecules elute at a later time. Okay, so class, that's it for our sec. Now, we will proceed with our nucleic acid classical analysis. So of course, with our nucleic acids, similar steps of isolation, purification, and assessment using gel electrophoresis, spectra, and PCR-based methods can be used. But before that, of course, for our classical analysis, that's your isolation and purification step. Always the wet lab techniques po, class ang ating isolation and purification strategies for nucleic acid. Starting with your cell lysis or homogenization, similar with our differential centrifugation strategy for proteins, diba? So next step, purification, and then your analysis. So it can be through electrophoretic experiments, spectro, PCR, or your any other methods. And lastly, if we still need to further examine the structure, the quantity, or the quality of our target biomolecule, of course, we need to properly store it, okay? So class, here are the reagents that you need in order to successfully isolate and purify your nucleic acid. In theory class, it's straightforward, but in the lab again, this is a dreadful isolation and purification step. So of course, prone ang undergrad students in repeating the cycle of your isolation and purification when upon assessment, hindi pure or hindi na-isolate successfully ang target nucleic acid. O diba? Sa mga nag-struggle sa thesis in your biochem lab, diba? Of course, this is really a very dreadful and tedious experiment. Let's check the reagents that we need to use. First, of course, to maintain the pH, it acts as a buffer. We have your tris HCl. It stabilizes the structure of your nucleic acids by neutralizing the negatively charged phosphate group in its structure, diba? So to form naman your ionic bonds with phosphate groups, still for stabilization ng structure ni DNA, we have your NaCl. Okay, so class, again, this is a common salt that we particularly use for nucleic acid isolation. Next, for SDS, this is mainly to denature your proteins. So class, in our nucleic acid isolation, we treat any other biomolecule as contaminants. That's why we need first to disrupt your cell membrane to denature your protein components. Of course, class, in your cell, lagi namang may proteins. That's why we still need to denature it. Next, for your EDTA, of course, class, from your Anachem, very familiar that EDTA is used for chelation. And similar with this, of course, in your biochem lab, it is also used for chelating your divalent ions, particularly your magnesium 2+. And class, remember, magnesium 2+ is a cofactor of majority of enzymes such as your nucleases. So, diba? So of course, very important si EDTA. And actually, SDS and EDTA already appeared in your board exam. Very straightforward definition type of question lang siya, class. So don't forget to memorize their specific functions. Okay, so going back, we also have your CTAB. Class, before I forgot, again, your SDS, it means your sodium diacetyl sulfate. EDTA means ethylene diamine tetraacetic acid. And CTAB, that is your cetyl trimethyl ammonium bromide. Class, please also find some time to remember or memorize their full name. Okay, sayang naman yung 1 point if the question is just a simple, straightforward, name your reagent type of question. Okay, so again, si CTAB is specific in precipitating your polysaccharides or in general, your carbohydrate components of your cells. Okay, so si phenol and chloroform, actually this is to fully denature your proteins and of course is used in separating the aqueous and organic phase molecules. Okay, so of course, again, this is a very familiar separation strategy that we discussed in your analytical chemistry. And lastly, for ethanol, again, class, this is used for further purification via washing. Ito na ang final touch, class. Again, it will precipitate out your nucleic acid finally, diba? And used for washing for further purification. O class, after we isolated and purify your nucleic acid, one main analysis strategy is your spectrophotometry. So of course, class, in order to quantify the purity of your nucleic acids, the DNA and RNA, we use their absorbance ratio at your 260/280 nanometer wavelength. O diba? So of course, there are interpretations here. Let's check. Ayan, class, diba, recall, your nucleic acids, they are capable of absorbing UV light at 260 nanometers. Okay, so we expect that in this wavelength, of course, there is a high absorbance. And for 280, of course, your ribose sugar, a component of your nucleic acid, can also contribute to the UV absorbance. So how do we interpret the result? Class, these are the only important or essential info that you need to know for the interpretation of result. So if around 1.8 ang ratio, so of course, you need to divide your 260 nm absorbance to your 280 nanometer absorbance. Again, going back, if it's 1.8, we are sure that our product or sample is pure DNA. All right? If it's 2.0, of course, we are sure that, of course, we have your pure RNA in our sample. So class, what's going to happen if there is a low ratio, meaning less than 1.8? So we have a presence of contaminants. So kapag low and high ratio, your sample is happy and contaminated, diba? So of course, for low ratio, it can either be your protein or phenol or other contaminants na pwede nating i-blame. All right? For high ratio or greater than 2.0, of course, your RNA can also contaminate your DNA sample. Okay? Or aside from that, there will be a buffer interference. Your tris HCl is also capable of contributing in your UV absorbance. Okay, so again, class, common contaminants, remember, your proteins, the phenol that we use in separating or separation process, your guanidine group, the solvents used in your purification strategy, or any residual reagents coming from your previous reaction or isolation strategy. Again, class, remember, your spectrophotometric determination of purity ng nucleic acids, 1.8, pure DNA, 2.0, pure RNA. Okay, so class, at this point, we are now going to move forward to a more advanced biochem lab techniques. Don't worry, relatively shorter ang discussion natin on this. And in order to start our discussion, we will start first with electrophoresis. So class, let's watch this short video in order for me to introduce to you our gel electrophoretic experiments.
Gel electrophoresis is a technique for separating DNA fragments according to their size. It is used in DNA fingerprinting. How does it work? First, a gel slab is prepared.
Class, of course, our typical gel we use is your agarose gel or your polyacrylamide gel. So later on, we will differentiate what are the samples that we can use for your agarose and your polyacrylamide.
Then, samples containing DNA fragments of different sizes are loaded into the wells of the gel slab. There are many molecular spaces in the gel slab. These spaces allow DNA fragments to pass through the gel. An electric field is applied to the gel slab. Since DNA fragments are negatively charged, they move towards the positive terminal of the electric field. DNA fragments of different sizes move across the gel at different speeds. The shorter the fragments, the faster they move.
So actually, class, the hardest part here in electrophoresis, same with IEF, is the sample introduction into your well. Because if hindi ka careful or wrong yung angle mo, of course, it will result to smearing ng bonds. So pangit siyang i-visualize if there are smearing of bonds. Okay, so let's go back.
Therefore, after a certain period of time, shorter fragments travel further than longer fragments in the gel slab. In this way, DNA fragments of different sizes are separated into bands. Since the DNA bands are invisible, they are usually stained with a special dye after electrophoresis, so that the bands can be seen.
Ayan, so class, again, to summarize your electrophoretic technique, here our separation is through your charged molecules in your electric field, and class, solely based po siya sa size ng molecules natin because the charge to mass ratio is uniform. Okay? So again, because of this uniformity in your charge to mass ratio, we can rely solely in your separation based on size. Class, again, for our gel type, it can be your agarose gel, which is suitable for your large molecules such as your nucleic acids and your polyacrylamide. Ayan. So for your smaller fragments of nucleic acids such as your RNA and your proteins. So for your proteins, we call the process SDS-Page. Okay? So before that, of course, for your nucleic acid separation, again, solely based on size, because class, I would like to emphasize the charge to mass ratio is again uniform. Alright, for protein separation, here we need your SDS first to denature the proteins because this gives your uniform negative charge in the structure of your protein, and hence we can also now solely base on size and their separation. And ang result, ayan, your biomolecules will be separated by molecular weight. O see, ayan class, again, the hardest part, ang pagload ng protein sample in your wells, and then afterwards, you will apply your current or your electric field where it will induce the separation of your different components. And here, ayan, here is the visualization of your bands. Ang ganda ng resulta, walang smearing, diba? So sana all class, ayan. For agarose gel electrophoresis, this is just a more detailed methodology in your age technique. Okay, again, here we can use your tris acetate EDTA or your TAE, diba? Or your tris borate EDTA in order to prepare your agarose gel. And afterwards, ayan, we just need to polymerize the gel before it is ready for DNA loading or sample loading in your wells. So class, again, afterwards, similar processes na lang. Electric field is applied, and then your DNA fragments will be separated based on size. Again, class, the smaller molecules will elute faster, so they are at the end front of our gel. Okay? So yung mga nasa unahan or your nearby your wells, of course, those are larger molecules. And lastly, you can stain the gel and visualize it under UV light, or ayan, you can proceed with the blotting. Okay? So here is your blotting techniques integrated with gel electrophoresis. Again, you can transfer your electrophoretic result to a membrane, so usually nitrocellulose membrane, and then we will use specific probes or antibodies for further identification of your specific sample. Well, let's see. So class, usually again, we use your nitrocellulose membrane. So class, here is the result coming from your electrophoretic experiment. So we will use your nitrocellulose membrane for your blotting and migration ng nucleic acids onto our nitrocellulose membrane. See class, this is actually a complex technique. So actually, I did not experience this in our laboratory. So here is our autoradiography of your visualized label bonds coming from your electrophoretic technique. Or sir, does this appear in the board exam? So actually class, ang important thing here that you should know is the different types of the blotting techniques lang talaga. So the southern, the northern, and the western, actually they have different target biomolecule. Okay? So I summarized it in your manual. So you can check it class, but here is the same summary, for our different blotting techniques. See, for our target sa southern, DNA. For your northern, RNA. So for both southern and northern, they are nucleic acids. That's why our probes are also nucleic acid. Okay? For our western blotting technique, class, specific siya sa protein, and that's why we use your primary antibody similar with your ELISA. Alright? Ang purpose again is to detect your specific biomolecule. For your southern, that's your DNA. For your northern naman class, again, si RNA is our main regulatory point for gene expression. That's why it's also used for measuring the gene expression levels through the quantity of your RNA in our sample. And lastly, for western again class, it determines the quantity or identifies the specific proteins in your sample. Class, for the sample prep, detection method and application, so of course, very straightforward na lang siya. Sample prep, we already discussed this from our protein and nucleic acid classical analysis. For detection method, almost all of them are through autoradiography, such as your X-ray film, chemiluminescence, or your infrared imaging system. So sir, what is the relevant application of these blotting techniques? Sa southern class, because it's DNA, it actually identifies gene mutation. O, diba? For northern, mRNA expression, very important in measuring gene expression levels. And lastly, for our western, it's to diagnose diseases. Actually, class 4, enzyme defects. For the quantification or lack of enzymes in our biological system, we use your western blotting technique. Okay, so sir, mag-mnemonics ka ba here? Yes po. O, diba? For our 3 blotting techniques, drop some real nonsense para win. O, diba? For your drop, of course, the DNA, some is for southern. So DNA for southern, RNA, real nonsense, northern. And lastly, your protein is for western. Drop some real nonsense para win. O, class, you can use the mnemonics, or if you have existing mnemonics, feel free to use use that. Okay, so class, ma-excite na kayo. We are now done with our blotting technique. So actually class, we are now left with 3 more techniques na lang po. Okay, so here is our third to the last technique, si PCR. So I think you are already familiar with this because in your or during the pandemic, the PCR has become famous because your RT-PCR is the main, di ba? Main technique to detect the presence or confirmatory test of your COVID-19. Let's see the typical processes for your PCR. First one, DNA extraction from our sample. O di ba yung swab test? Of course, that's a type of your sample extraction or sample collection, and then we will extract your DNA from that sample. Second, amplification. So class, in our PCR equipment, this process is automated. So we will just put your PCR tube in your PCR equipment and the amplification step is an automated process. As you can see here, it is a 3-step process. First, upon introduction of your DNA sample, it will be denatured first at your 94 to 98 degree Celsius. And then we will start the annealing process with your Taq polymerase. So later on, we will enumerate the components of your PCR tube. So this one class, of course, hindi lang DNA ang laman yan. Of course, you have your other PCR components. And lastly, elongation. Ayan. And afterwards, your cycle will just repeat in order to exponentially double the concentration of your DNA or initial DNA sample. Diba? And lastly, we can analyze it through your electrophoretic technique. Class, here is the list of the components of your PCR tube. Again, hindi mawawala ang ating DNA template. So this contains the target sequence to be amplified. Sir, yan po ba yung DNA sample? Yes. Diba sa swab test in your sample collection, of course, afterwards, we will treat that sample in order to purify and isolate the DNA of interest. Next, we have your primers. From your application, we know that your primers flank our start of your elongation process. So here, the primers attach to the beginning and end. That's the meaning of flank. So your short single-stranded DNA will flank your target DNA region. So afterwards, we have your DNTPs. Of course, these are the building blocks, your 4 nucleotides for new DNA strand synthesis. Because class, wala tayong source ng DNTPs inside your PCR tube. And here, the one responsible in elongation, your Taq polymerase. Diba recall, we discussed your Taq as one of your extremophile enzyme. That's why it's very suitable in PCR at high temperature condition because it is a heat-stable enzyme that is used for elongation or strand extension. And additionally, class, don't forget, we need to maintain, your optimal pH and salt conditions. That's why we use buffer such as your TRIS-HCl and ETA. And lastly, we need your cofactor, magnesium 2+ for your polymerase or your Taq polymerase activity. Class, ayan ang ating PCR tube with its components. So see, again, your DNA template, the primers, your DNTPs, so particularly your 4 DNTPs, your Taq-pol, and your buffer along with our magnesium cofactor. Diba class, please remember the components of your PCR. Here naman, ang commonly na tinatanong is your Taq polymerase. So which of the following is the enzyme mainly used in the elongation step of your PCR? That is your Taq polymerase. So class, to end our discussion here on PCR pa lang, here is our Did You Know segment. So class, this is in relation to your COVID-19 pandemic. Or recall, PCR or in particular, RT-PCR is the frontline tool against SARS-CoV-2. Diba? A single viral RNA strand can actually be detected by your RT-PCR because it can amplify your initial RNA molecule into billions of copies within hours. Class, remember, our RT means here your reverse transcription. So class, ang ating sample, initial sample is your RNA molecule. So again, RT-PCR is critical or was critical in diagnosing COVID-19 by converting your viral RNA from your SARS-CoV-2 virus into your complementary DNA. And of course, afterwards, it will be amplified for detection. So during the pandemic, actually, the PCR method became the gold standard for identifying infections early. Actually, class ngayon, meron ng wide or extensive application of PCR sa clinical settings. So it can also detect your HIV or it can also quantify it. Other sexually transmitted diseases, your gonorrhea, your CTNG, of course, and many other diseases. Diba, the birth of your PCR came during the pandemic. Alright, so class, with our second to the last technique, we have your ELISA, your enzyme-linked immune sorbent assay. So this is for your proteins, antibodies, or hormones quantification and detection. So class, well, simply, this is very specific for your proteins. So let's see. Ayan. For our first step, of course, as you can see here, your antigen antibody is immobilized on a microplate well. So depending on the type of ELISA, we can use your antigen or antibody. Next, we will block the non-specific binding sites to prevent background signals. So those are located in your sample. So we need to block it for our quantification strategy to be accurate. Third one, sample addition. So here your sample is added to wells where the target molecule is added and binds to the coated component. Diba? For instance, here you have your antibody. Number 4, detection. Antibody is added to bind the target molecule. So class, in our next step, here is our main substrate reaction wherein your enzyme will catalyze a color change and this will be detected through your spectro measurement. So color intensity is usually measured using a spectrophotometer. So actually class, in our second step, we use a HRPO or your horseradish peroxidase in order to induce the coloring of our sample. Diba? So of course here again, we have your enzymatic color reaction proportional to your antibody. So here we use your TMB substrate to couple with your HRPO in order to get your color change. Clear? Ayan, so class here, I would like to emphasize the different types of ELISA. Ayan, we have your direct, indirect, your sandwich, and competitive. So class, they only differ in their sensitivity and applicability on your biomolecules. Let's see. Ayan, indirect, it detects your antigen using a single enzyme-linked primary antibody conjugate. Class, you can see your silver nanoparticle is used to immobilize your antibody conjugate in our well. Okay, so class, let's go to our indirect ELISA. And here, we use 2 different antibodies. One is unlabeled and one is enzyme-linked secondary antibody for amplification ng signal natin upon color change. And of course, class, similar substrate will be used to induce your color change. And here, for our third one, sandwich ELISA, of course, from the name itself, we will sandwich your antigen via the use of 2 antibodies. One is capture and one is for detection. So here's a detection antibody will be the site of binding of our substrate to induce color change. And of course, our measurement is through spectrophotometer or spectrophotometry. And lastly, for your competitive ELISA, ayan, almost similar lang siya with indirect, but here, the signal that we actually generate is inversely proportional to the antigen amount. So class, the reason there is that your antigen actually competes with the labeled antigen for binding kasi wala siyang inhibitor antigen. Okay? So class, I hope that's clear. Ang important lang in your ELISA is your direct and sandwich actually. Also in terms of sensitivity, what are their differences? Class, from your direct all the way to your competitive ELISA, sensitivity and specificity increases. So meaning, sir, less sensitive to direct? Yes po. While the competitive, of course, because of its high specificity and sensitivity similar with your sandwich, so both of them have high specificity and sensitivity, they are useful for small molecules. Okay, so class, again, I would like to emphasize the important type of ELISA here is sandwich and your direct ELISA. Let's proceed with our next and last technique for our crash course 10, our recombinant DNA technology. Class, you can now see the light now. Before we finish our discussion, let's proceed first here in your RDT or your recombinant DNA technology. Alright class, in addition, your recombinant DNA technology is actually the pioneer technique or a pioneer technique in your genetic engineering industry. So later we have a Did You Know segment on the production of commercial insulin through your recombinant DNA technology. Class, again, this technique allows the isolation, modification, insertion of your specific genes into host organisms in order to produce our desired traits or proteins or hormones. Let's see the overview of your recombinant DNA technology. Diba you are curious with your plasmid DNA? Again class, it has a very important part in our recombinant DNA technology. Remember, it's different from your chromosomal prokaryotic DNA because your plasmid DNA only carries extra genes. That's why it's very suitable as our vector for recombinant DNA technology. Class, aside from your plasmid vector, we also need your restriction enzymes in order to cleave your original plasmid vector, your ligase, your PCR, your vectors, and your electrophoresis. So the PCR class and electrophoresis, they are mainly used in your screening and purification. Alright? For the application of recombinant DNA technology, we have your cloning and mainly the protein production or your therapeutic protein production, such as your insulin and many other vaccines. Let's see the steps and processes under your recombinant DNA technology. Let's see. Ayan. First thing to do, we isolate the gene from your plasmid vector. So here, we will use our restriction enzymes to cut your desired gene. Ayan siya class, diba? So of course, we will also cleave your plasmid vector via your restriction enzyme. Second, insertion into your vector. So the DNA fragment, or we also call it your insert, okay? It will be inserted to your plasmid using ligase. And then, transformation will occur wherein the recombinant vector class, here it is, will be introduced to your host cells or your bacterial hosts, such as your E. coli. So class, si E. coli as well was used in your insulin production. And fourth, ayan, for expression, your host cell must successfully express the gene, thereby producing your desired protein or trait or hormone. And lastly, in order to confirm the success of our recombination, we need to screen and purify our host cells in order to confirm the recombinant DNA and its gene. Ayan. So class, aside from that, of course, here in your recombinant DNA technology, very crucial that our vector, particularly for instance, your plasmid vector is a very good cloning vector. And of course, may criteria for judging po tayo for your good cloning vector. Class, remember, this 6 traits of your or 6 features of your good cloning vector. In your board exam, it always appear annually. And ang answer dyan class, laging all of the above. But again, please be careful in analyzing your choices there. Again class, for your good cloning vector, it should be a small size vector. There is an origin of replication. There is a selectable marker. It has specific restriction sites, high copy number, and stable maintenance. So of course, class, in general, your vector must be easy to manipulate and replicate inside host cells. That's why it should also allow autonomous replication of your vector in host cells. Kaya dapat meron siyang RE and restriction size. Okay? So class, now we are ending our discussion with our Did You Know segment and our 2 specific sample problems. Let's see. Ayan. So again, did you know that the first genetically engineered drug approved for human use was insulin? So this is through your recombinant DNA technology. Let's read. Again, your scientists inserted the human insulin gene into your bacterial host, your E. coli. So in order to produce your human insulin efficiently. So this breakthrough actually transformed your diabetes care and launched the era of biopharma, making insulin safer, more accessible, and widely available. Class, diba successful ang gene expression of your human insulin? Here, they use your E. coli as their bacterial host in order to use their plasmid DNA. And of course, upon successful recombination, it underwent large-scale fermentation to mass produce your insulin. And that's the history of our commercial insulin. Class, now let's proceed in answering your sample problem. Again, this is also given in your manual. Let's check. In Western blotting, proteins are first separated by gel electrophoresis and then transferred to a membrane for detection. Class, the question is, what specific molecule is used to bind to the target protein? Is it your RNA probe, your DNA probe, your antibody, or enzyme? Class, recall, your Western blotting, our target is your proteins. So class, for our probe or the binding site, it shouldn't be a nucleic acid. So obviously, RNA probe or DNA probe must be eliminated. And from our discussion, remember, very big si enzyme, diba? So here, we use your antibody in Western blotting as our probe to specifically bind your target protein. See, again, for our Western blotting, our probe is your antibody in order to produce your autoradiograph. Okay, so now class, for our last sample problem. Here it is. In your recombinant DNA technology, what is the key tool used to cut DNA at specific sequences to facilitate the insertion of a gene into your vector? Class, very fresh ang discussion natin here. Is it DNA polymerase? No, diba? So your DNA polymerase does not participate in the cleaving reaction. Is it ligase? Well, possibly. Is it restriction? Is it the restriction enzyme? Let's see, class. Or is it the reverse transcriptase? So again, class, here is that specific enzyme that will cut your DNA at specific sequences. And we know that that is your restriction enzyme, not your ligase and not your reverse transcriptase. So reverse transcriptase actually is in your RT-PCR. So in particular, restriction enzymes act as molecular scissors to cut DNA at specific sequences, thereby enabling the isolation of genes for insertion into vectors. All right, so class, your favorite slide is here. Ayan. So this one is the summary of our crash course number 10. O, diba? So class, remember for Chrome, purpose mainly is for separation and purification. From your anachem, this is based on size, charge, affinity in your ion exchange, or affinity chrom, or size exclusion chromatography. Second, we have your protein analysis and actually class also your nucleic acid analysis for isolation, separation, or purification. O, diba? We have an extensive discussion on these two types of analysis. For protein analysis, we discussed your ammonium sulfate precipitation, your isoelectric focusing, your chemical digestion, your differential centrifugation, and lastly your size exclusion chrom. For nucleic acid analysis, on the other hand, we mainly discuss your spectrophotometric determination of the A260 and A280 ratio absorbance. Of course, we also discussed the techniques electrophoresis and PCR. Class, again, your electrophoresis and PCR are mainly for large biomolecules. For electrophoresis, size-based separation. For your PCR, mainly for amplification for detection of certain DNA sequences in our sample. Please remember our PCR components, your primers, the NTPs, the very crucial and important Taq polymerase enzyme, and your thermal cycling. Okay, so class, I hope that's clear. Second to the last is your ELISA that is specific for your protein or antibody quantification. And class, remember, our key feature here, is the specificity of the different types of ELISA, as well as the colorimetric detection via enzyme-linked antibodies where the measurements are done through spectrophotometric determination as well. Diba? Gamit na gamit ang spectro from your anachem. That's why it's very important that you know your Beer-Lambert's law. And finally, for RDT or your recombinant DNA tech, of course, main purpose for gene cloning or for your therapeutic protein production and expression. And remember, our RDT tools, C vectors, primarily ang plasmid DNA, we have your restriction enzymes, your ligase, diba? And of course, in your transformation, we also need to screen and purify your host cells that successfully express our genes of interest. All right, yay! So we are done with our whole discussion ng crash courses 1 to 10. And to end our video here, class, ayan, let's have a brief review checklist. First with central dogma. First one, we identified the central dogma processes and the direction of information flow, in particular from DNA to RNA to protein. And of course, similar with our discussion on your metabolic pathways, we also discussed the subcellular location of your central dogma processes. Recall, in your eukaryotic cells, your replication, your transcription, they occur in your nucleus. And lastly, for translation, it occurs in your cytosol, in particular sa ribosome, class, diba? Thirdly, we also recall your key biomolecules involved in each process. O class, let's take some time to revisit our discussion on the three processes under central dogma. And lastly, ang impact ng mutations, particularly in your replication, and then the domino effect in your transcription and translation. Okay, so class, for your lab techniques, ayan, we have 4 review checklists. First one, we identified, diba? We discussed each lab technique's purpose and application. We also match each technique to their target molecule, diba? For DNA, RNA, or protein in your blotting techniques, for your PCR, for the DNA amplification, ELISA, for protein quantification, and of course, your recombinant DNA technology for DNA manipulation. Okay? Thirdly, we also successfully recall the key components or conditions, diba? Key components ng PCR, conditions of your recombinant DNA technology, and the electrophoretic technique. And lastly, ayan, class, don't forget our results interpretation solely via spectrophotometry. Very easy ang interpretation ng results here in your biochem techniques. And that's it for our biochem crash courses. For this video, for the past 3 videos, we discussed central dogma that involves your DNA organization, your replication, your transcription, translation, and finally, mutation. And in this video class in particular, ayan, the characterization and biochem lab techniques. And now we arrive with our final slide for biochem. Class, are you ready to answer our last practice test? So class, again, please answer your last practice test that is 10 items for 15 minutes. So I will see you on our discussion of your answers in your PT 1 to 10. So later on that discussion, I will let you tally your scores from your PT 1 to 10. So hopefully class, maganda ang performance nyo in our biochem subtest. So class, I would like to thank all of you for listening and attending our asynchronous online discussion of biochem crash courses. So class, for now, you can take your break, your sanity break, hydration break, and of course, your well-deserved break, diba? So that's it for our biochem crash course. Bye class, and see you in our PT discussion. Bye!